Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. As you guys know, we're in the coronavirus shutdown. This is supposed to be a face-to-face class, but we have switched to online. So these videos are being done as quickly as possible to keep my students moving. They're all one-take videos, and if you hear any barking or beeping in the background or kids or anything, please forgive me. I'm doing this at home, doing the best we can to get through this difficult situation. Also, the screen is kind of dark, but what I found is if I brighten it up the way that it looks good when I'm staring at my computer, you can't see the writing on the board, and ultimately that's what we're here for. Now, today's lecture is going to be over the nervous system, and this is going to start for my students. These lectures are intended for the students taking my courses at Del Mar College. If anyone else out there in YouTube land stumbles across them and finds them helpful, hey, let me know. I plan on redoing all these videos eventually in a much more slick and professionally done, professionally edited format. But right now, I'm getting through this. So this lecture is for my Human Anatomy and Physiology 1 course, Biology 2401. We divide our course into 6 lecture exams. And although this material should have been on the last lecture test, I realized I left it off, so I'm just going to throw it on test 6. So this is sort of the first lecture when we start talking about the spinal cord and moving on through the end of our note set. For those of you in my class, we're going to follow along in the note set. Now, what I left off is I didn't talk too much about the ventricles and the meninges, and they're on page 73 of your note set. We're going to do a little bit of page 73 and 74, and then we're going to jump all the way to the spinal cord, okay? It's only because I left some of this out, but I think it also fits in with the spinal cord. So if we were looking at the brain from somewhat of a, let's say an anterior view, okay? You would have the medulla continuous with the spinal cord down here. You would have the pons, and then we would have an area called the midbrain. And then sitting up here would be the diencephalon coming across like this, okay? And then the cerebrum would be all the way up here. We're not going to worry about that. As you guys should know, there's a structure out here called the putamen, the little bean-shaped structure. And then we have a little bit of cerebral cortex on this, which is called the insula. Now, we have an area in here that's kind of a bunch of wires going through called the internal capsule. We would have that on both sides. I'm just going to put IC for internal capsule. Now, we have a structure that starts here and wraps up and goes all the way around underneath like a C-shaped structure clamped onto something, and that would be our caudate nucleus. I'm going to erase that arrow because now that you understand that. So our caudate nucleus kind of wraps here. And then all of this across here would be thalamus. And of course, the tip of the thalamus would be the hypothalamus. But imagine if we cut the brain, cutting in a coronal section here. Now, sitting right on top of the thalamus is a rounded structure called the fornix. There's a structure up here called the corpus callosum, and there's some cortex over here. And we have the septum pellucidum, which is a membrane that connects the corpus callosum to the fornix. Now, if we were looking at this from a lateral view, we would have the medulla and the spinal cord. We'd have the pons. We'd have the midbrain. Back here would be the cerebellum. And then we'd have the diencephalon, like the thalamus with that intermediate mass or interthalamic adhesion, the hypothalamus. Our fornix would be sitting here. The corpus callosum would arch over like this, and we'd have that septum pellucidum here, okay? So we're going to look at this from an anterior and a mid-sagittal section, okay? I'm going to put the ventricles in blue. There's a ventricle on either side of the septum pellucidum here. And this ventricle actually kind of wraps around just like the caudate nucleus does, okay? And each one of these is called a lateral ventricle. And the lateral ventricles would be sitting right here on either side of the septum pellucidum, and they would wrap around something like this, okay? So this would be your lateral ventricle. They actually come to a little point back here. So they're sitting on either side of the septum pellucidum. Just like I'm sitting in a room, on the other side of this wall is another room. The wall would represent the septum pellucidum. The spaces would be the 2 lateral ventricles. Now, the lateral ventricles drain through a little tiny opening or hole at the top of the thalamus into the middle of the thalamus. So there should be a little connection right here on your models called the interventricular foramen. And then the third ventricle sits right over the thalamus like this and actually runs down into the hypothalamus like that. So fluid would flow around the lateral ventricle through the interventricular foramen and then fill the third ventricle. Running back here, if this is all lateral ventricle filled with cerebrospinal fluid, then back, I'm sorry, didn't do that in the right spot. Right about here, there would be a little channel running through the midbrain that opens up into a fourth ventricle down here, and then continues as the central canal of the spinal cord. So the central canal of the spinal cord is connected to the fourth ventricle. That would be your third ventricle. So the cerebrospinal fluid will circulate through here, through the cerebral aqueduct, and since it goes through the mesencephalon, which is the midbrain, it's also called the mesencephalic aqueduct, or the aqueduct of Sylvius, after a guy who described it. Comes through here into the fourth ventricle, and then down the central canal. So your third ventricle would be sitting right here in the middle of the thalamus. The mesencephalic aqueduct or cerebral aqueduct would be here, and on the back side of the pons, it would branch out like this into this little diamond shape where the fourth ventricle is. So from an anterior view, we can see the ventricles here. From a mid-sagittal section, you can see them there. Now, that's not the prettiest drawing, but you know, you got pictures in the book and you got other things to look at, the models from lab. What I want to get across to you is a couple of things. First and foremost, sitting at the floor of the lateral ventricles, right where the caudate nucleus is, there's a series of blood vessels here. There's some blood vessels right on the top edge of the fornix as well. And on the the bottom edge of the fornix as well. And there's some blood vessels all the way down here in part of the fourth ventricle. Those blood vessels that are sitting in these areas are called the choroid plexus. And as you know, the choroid plexus is a specialized network of capillaries. I talked about this in a previous lecture when we talked about the formation of cerebrospinal fluid and how we have to have specialized blood vessels to filter them, because neurons are susceptible to all the toxins and trash that other cells could dump into the bloodstream. If any of it gets into your brain before it's filtered, it can be harmful to neurons. They're the biggest wimps of the human body. So we have these specialized blood vessels in all four ventricles called the choroid plexus, which filters your blood and forms the cerebrospinal fluid. And, of course, the cerebrospinal fluid is going to flow down the central canal of the spinal cord here. Now, not only does it flow in the center of everything, it also comes out and flows around everything. If we were to look at the brain and the spinal cord, I'm going to do a smaller drawing over here just to illustrate something. Our brain and our spinal cord have a series of membranes that surround them. They're similar to what we consider to be the visceral and parietal pleura, those serous membranes, but they're incredibly different. They're called the meninges. You've heard of meningitis, inflammation of the meninges. Well, if it surrounds the brain, they're called cranial meninges. If they surround the spinal cord, they're called spinal meninges. They are the exact same membranes. There are three layers. The outermost layer, which is attached to bone, would be called the dura mater. I'm just going to put dura up there, but we use the term mater, which is like alma mater, it means mother. Dura means tough, like durable. It is the hardest one to cut. It's the toughest of the three. Now, the second layer sits underneath that, and there's a bit of a gap here. And the gap is actually a little bit more pronounced in the spinal meninges, but I'm going to exaggerate it some just to illustrate something to you. The next layer, which runs underneath the dura mater, is called the arachnoid layer, or the arachnoid mater. Arachnoid gets its name from the fact that it has all these little spider web-like structures that are supporting it and propping it up like this. Looks like a bunch of cobwebs in there, and arachnids are spiders. So they called it the arachnoid mater. The deepest of the three layers actually lies against the surface of the brain and the spinal cord, and it's called the pia mater. Pia means softer, durable, softer or nice, and the pia mater is right up against the surface of the brain and the spinal cord. There are some gaps between these layers. The space between the dura mater and the arachnoid is called the subdural space. Subdural because it's below the dura mater. The area below the arachnoid mater is called the subarachnoid space. And if we were looking at the cerebral cortex, sitting up here like this, those meninges, I'm just going to draw one color for the dura mater, those meninges would fold down in between here and wrap all the way around the brain and envelop it. This fold between the two cerebral hemispheres is called the falx cerebri. What's really important about the falx cerebri is it has two blood vessels in it. One way up here, called the superior sagittal sinus, and then one down here called the inferior sagittal sinus. They're kind of embedded in that layer. And one last detail. These little structures that are supporting the arachnoid mater and holding it up in here are called arachnoid granulations. They're little granular looking structures. The arachnoid granulations actually have some little blood vessels in them. One of the things that happens, and the reason the meninges become important, for a number of reasons they're important, but one of them is there's two openings at the fourth ventricle here. I'm going to use the light blue color here. But those openings would drain some of the fluid outside the central canal in the fourth ventricle and would drain it into the subarachnoid space. And so the subarachnoid space is filled with cerebrospinal fluid. I'll put it in green here. And cerebrospinal fluid flows all around the outside of the brain and spinal cord as well. It comes out of these two lateral apertures. It flows around the brain and the spinal cord and through the brain and spinal cord, and then it will be reabsorbed through the arachnoid granulations into the superior sagittal sinus. So it goes into these arachnoid granulations and then will be reabsorbed into the superior sagittal sinus where it joins our blood. That would be the dirty cerebrospinal fluid. Cerebrospinal fluid has three major functions. And all of this is listed on page 73. It talks about the flow of cerebrospinal fluid through the ventricles. It tells you the order of structures. You need to know that paragraph, okay? It describes the meninges. You should be familiar with those. And they'll be on my worksheet as well. Page 74 talks a little bit more about cerebrospinal fluid and the order of the structures through which it flows. It has three major functions. One is it's delivering nutrients and ions to and glucose and oxygen and other valuable substances to our neurons. It's feeding them, so to speak. Second function is as they dump their waste into it, it washes away all the waste and debris. A third major function is it acts as a shock absorber because it completely envelops the brain. Then if your brain is sloshing around in your skull like you're shaking your head no, it's not slamming into the wall. The fluid is hitting the wall and bouncing off, and it provides a little bit of a cushioning to the brain. Obviously, if you take a severe blow to the head, it cannot suffice for that. There's not enough of it, and then your brain would bounce around inside the skull and become bruised, which when we have a hematoma, broken blood vessels, and bruising on the brain, that's what we call a concussion. Anytime you break the blood vessels and have bruising, that means there's lack of oxygen flow to that part of the brain. It can be rather severe. That's why you hear all about concussions in football and other sports and things being a big deal. Nonetheless, that should cover the cerebrospinal fluid. It should cover the ventricles. I wanted to give you sort of an introduction so that you can see where it is. I wanted to talk about the main structures. Again, they're all in the note set, and there'll be a worksheet coming out for you guys. So this will be part of lecture test 6 if you're in my class, the last series of quizzes and things that we'll be taking. So thanks for watching. Hope you learned something. Hope you had as much fun as I did. Okay? See you on the flip side.